Okay, so the topic for today is going to be dynamic resting state fMRI. I think there's been more than one lecture on just functional connectivity for resting state fMRI. So today we're going to go into this extra aspect of it called dynamics, and I'll explain in a second what we mean by that. So hopefully today what I'll do is explain you a little bit what we mean by connectivity dynamics, talk a little bit about the original observations of this phenomenon, the kind of studies that started to get people interested into looking into this a little bit more. Like with anything that you see in fMRI data, it's always good to be cautious and think, is this something that it's telling me something about the brain, or it might be more of an artifact in my data, or something that I should look at with a lot of detail before I can make any big interpretation of it. So I will go through a series of studies that hopefully point towards the direction that this dynamic behavior is something that we should be interested in looking at. And then I'll move into a little bit of comments on methodology, because this is kind of a new area of research. So methods are evolving continuously, and there are a lot of options, and a lot of them are still under development, or we don't really know whether which one is the best way to actually capture dynamics that are either cognitively or biologically interesting in these signals. So just to make sure that we're all on the same page, most of the studies that you see in functional connectivity, what happens is you acquire data for 6-10 minutes. In this case, we went a little bit crazy, 60 minutes, but that doesn't matter. And you do your seed-based analysis or your ICA decomposition, taking the whole time series and applying it to the whole time series at once. The idea being that what you're looking at are temporally stationary things, and therefore it makes sense to look at the whole data because you get the best chance of looking at what's going on. But as I will show you in a bit, people started to think, well, what if we, and okay, so here let's say we have a seed in the posterior cingulate cortex, and this would be just basically a map of connectivity of the time series in this area to the rest of the brain, and you get something that looks like the default mode network. But nothing stops us from actually saying, okay, what happened if I just look at the chunk of the data? And here I'm just looking at 5 minutes because I have 60, but the same applies should I have 10 minutes and I decide to look to the first 2 minutes. What we will see usually is that if we do that, the parents of connectivity for exactly the same seed region, just a different instance of our scans, may look quite different. And I will show you study after study that says that actually they differ significantly from what you see when you look at the whole time series as a single unit. So again, this is a field in which there is tons of studies, and it has exploded quite a bit in later years. So by no means what I will cover here is an extensive review of everything, and most likely by no means is the latest research available. So those are good places to go for additional information. This one starts to be a little bit outdated, but this one is pretty recent, and it's a really good review of both methods and the research. It has more of an emphasis in methods potentially, but might be a really good start point. Also, a special issue on just this topic of dynamic connectivity, it's been published these days in Neuroimage. As far as I know, those are the papers that are already out, but there will be more coming soon. So this is also a good place to look for the latest developments in some of the topics related to dynamic connectivity. As the talks will be later available, that way you have all these references. Okay, so let's go back to some of perhaps one of the first studies that mentioned this idea of temporal dynamics in connectivity. This is work by Katie Chang and Gary Glover. And what they said in that original study is that most studies of resting state functional connectivity employ methods that assume temporal stationarity, what I was just saying, such as correlation or data-driven decompositions that they are performed across the whole duration of the scan. However, there is evidence, looking at the data, that functional connectivity may exhibit changes within the scale of seconds to minutes, within that scale of what a single scan is. Here what we have is 2 time series, one for the posterior cingulate cortex in blue and another one somewhere else. And just by looking at it, one could argue that around here, both of them seem to go together. So most likely they're positively correlated with each other. But it's clear that here there is quite a long period of time. It's not just one data point, but around 100 seconds in which both time series go completely anti-correlated and everything in between, most likely. So one way to look at that is to actually get estimation of the evolution of the connectivity between these 2 time series. And One of the most common ways to do that is a sliding window, which I will go into a lot of details in the third part of this talk. But basically, you pick a window of time, you compute the correlation value for that, and you put it here. You move the window a little bit, you compute the correlation for that window, and you put it. And you come with this new time series, that is the time series of how these two original ball time series correlate with each other as time evolves. Of course, tons of decisions here, the size of the window, the shape of the window, the overlap between windows, and we'll talk about those things in a second. But just to show here that what we kind of observe here, we can get it quantitatively, and we can see that these two time series seem to go together at the beginning, then they seem to go against each other, and then they seem to come back. So that was one of the original observations. They went a little bit further, and what they realized is that the amount of variability on these traces of dynamic connectivity, not per se in the original time series, but on how ROIs connect to each other, had some sort of a spatial distribution. It wasn't at random. No? We can see in this case they're looking at the variability of those traces of dynamic connectivity against the PCC, and we can see there are some areas where that connectivity is much more variable, much more volatile than in some other regions. I also think this is a great study because even from the beginning, they started to talk about all the problems that in the last 5-6 years we're still struggling with. So they say it's still unclear whether the observed coherence or variability can be attributed to residual noise or modulation of cognitive state. If this is the case, very good for neuroscience and for clinical applications. If this is the case, then we shouldn't be spending that much time on this phenomenon. And the present result illustrates that resting state connectivity might not be static, and it may prove valuable to consider these measures. If this is where we are, and their original observation, because they found this structure and a few other results points towards that direction, they say that this might be a new dimension to look into our resting state data. Another way to look at this, and you will see many papers that do use this type of representation, you may have a parcellation of the brain, say 100 ROIs, 200 ROIs, and you compute the connectivity of every single ROI to every other ROI, and you come with a matrix of connectivity. If you do that with the whole duration of the time series, you just have one of those matrices per scan. Those matrices are symmetric across the diagonal, so you can always take the top part and extend it as a vector, and that's what become a column in this dynamic connectivity matrix. If we have running windows, what we can do is we can do that for every window and then kind of concatenate them together. So in a matrix like this, what you have is a given row is just a given connection, like ROI 20 to ROI 60. And what you have for that row is the evolution of the connectivity across time. And what this shows you is that there is definitely an evolution here. It's not flat. There are moments in which this ROI seems to be strongly connecting, then they go down, then a subset of them come up. And at any point, we can pick any of those columns and bring it back to a connectivity matrix to actually look what is the configuration of connectivity within that window. So some of the studies decide to look at this dynamic behavior was originally observed in awake humans, but they also decided to see whether it was present in anesthetized animals. No? The idea potentially being, well, if it's present also in anesthetized animals, then it's harder to make that direct connection with cognition. And in the original studies, they saw that there was still dynamic behavior present, but the more that people have been looking at this, what they realize is that the amount of dynamic behavior that is present goes down with increasing level of anesthesia. And there are a couple of papers out there showing this both for animals and for humans. So that tells you that, not all the dynamics can be attributed to ongoing cognition, because definitely there are residual dynamics even in unconscious states, but a chunk of it is possible that that's the case. Here in our group, we decided to look at, in the original study, we saw there is this kind of spatial patterns of the variability, but that was just constrained to the connectivity from the PCC to everywhere else in the brain. So we decided to look if we were to dig into this dynamic behavior, whether we would find connections that tend to be more stable across time and connections that tend to be more variable across time, with that behavior being common across all the subjects, and see if there was some meaningful spatial structure to this contrast of most stable versus least stable. And we found that most stable connections tend to happen between symmetric interhemispheric homolog regions, but also that they only account for 32% of intra-network connections. What that means is most of the networks that we're used to see during resting state, a static resting state, the full mode network model, they break down quite often because not all the connections are stable and are on all the time. And the most strongly connected one seems to happen, a stable one seems to happen between sensory motor networks. On the opposite end of the spectrum, what we found is that the most variable connections tend to correspond primarily to internetwork interhemispheric connections, mostly between frontoparietal and occipital regions. Doing a completely different methodological approach, the group of Elena Allen found similar results with these areas that they call the zone of instability that you can see are part of the full mode network, are parietal regions and frontal regions being the ones that show more variability within the duration of the scan. If you compare this with previous studies, looking at which are the connections that are more variable across subjects, they seem to overlap. And perhaps more importantly, or something curious to think about is that these regions that we see here and are similar to the ones I just showed you are the regions that have evolved later in time from an evolutionary point of view in the brain. And those are the regions that tend to be involved in higher order cognitive functions. So it kind of fits the story that maybe those regions that have higher order cognitive functions, that have more variable functions that are not so strictly defined as primary motor or primary visual, tend to have connections that are not only more variable across subjects, but also within the duration of a given scan, they tend to vary more. And again, all I'm trying to give you here is hints at studies that seem to point to the direction there is some value to be looked into this dynamic connectivity. This is another study that I find very interesting, in which they look at connectivity and they have some parcellation of the brain, and they decide to divide their connections in three types. Interhemispheric connections between homologous ROIs, things like this. Interhemispheric connections between non-homologous ROIs, and intrahemispheric connections. And they did this study both looking at humans and macaque, and the first result that we can see here, which is a little bit of a confirmation of our own original results, is that most stable connections tend to be those that are the ones that are interhemispheric connections between homologous ROIs. That is nice, but I think what's very nice in this study is that they decide to go one step further. They focus on these interhemispheric connections, and they try to look at the profile, looking from an anterior-posterior direction in terms of the stability of the connections, how variable they are, and a series of characteristics of the anatomical characteristics of the connections. And they found an interesting correlation between the diameter of the axons in the anterior-posterior direction and the profile of variabilities, showing a first link between structural connectivity as a potential surrogate or explanation to a given extent of the variability that we see functionally. Of course, in all these studies, and I will get much more into this later, if confounds like head motion, whatever it is, are a problem with resting state from a stationary point of view, they become even larger when you're looking at dynamics because you have shorter temporal windows, so any outlier that may come from an artifact may just look like, you got a change in connectivity. So in this particular case, they were interested in at least showing that the stability wasn't just a function of the distance. And what you see here is the stability of the connections as a function of distance, and you can see that the most stable ones, it's not like they are all clustered around here. They cover the whole spectrum of distances that they had on the study, and that's the case both for the human data and for the macaque data. Then another thing that has gained a lot of attention is this idea of functional connectivity states, because that was another observation made originally by Elena Allen, in which what they saw is that when you look at this matrix of connectivity on a window basis, if you were to take your time series, divide it in 100 windows, you have 100 matrices, what you start to realize that yes, this configuration changed a lot from one to another. There seem to be these recurrent short-term whole brain connectivity patterns that are not only present in a given data set, but that are common across subjects. And the term coined for that has been functional connectivity states. So the idea here, you have your time series, you do a window correlation. For each window, you get a connectivity matrix, and then you do some aggregate across the subjects and do use some type of clustering based on the spatial, well, in this case it's not spatial, but based on the similarity of this connectivity matrix, you find some representative ones and the moments at which the window connectivity looks more like those representatives. This is a great example also to see, here I have data, here I throw it methods, and it gets me something, and it's always going to give me something, because k-means has to give me clusters. It's a hard clustering algorithm, so if I tell him give me 7 clusters, it will give me 7 clusters. So it's important to then look or try to make sure that that structure that I imposed from my methods point of view was truly there in the data, okay? By that, there are a lot of tools that you can do to do some type of validation, but you can also have some a priori hypothesis or try to look and interpret what this might mean. This is something really hard to do when you have resting state data, but I think they did a good job in their case. So basically in their study, they combined data from around 400 subjects. They did their whole pipeline of analysis, and they came to the conclusion that there was 7 states, okay, with each of these states having this particular configuration. And then the other thing they decided to look at, because it's hard to know during rest what are subjects doing, whether they're all doing the same or not. The other thing they decided to look at, it was at the presence of the state as a function of time. And this particular state, they saw that it was very clear that the later in the scan they would look for this state, the more than this state appear. And they also saw that this state has 2 important characteristics. One of them is a strong anti-correlation between subcortical regions and sensory motor cortex, and also kind of a disruption of the default mode network. You can see this is the box that would correspond to the connectivity of all the ROIs within the default mode network. It seems to be broken compared to many of the other states. And these 2 things seems to be signatures of drowsiness. It kind of makes sense that the longer subjects are in the scanner, the more likely they will be drowsy. So that was one of the potential ways of, making sure that what you're seeing in the data may make sense. Is that strong enough to claim that this is all real? Potentially not, but it's little by little. This is science. Okay, so just a few initial conclusions. Functional connectivity exhibits a rich dynamic behavior at the scale of minutes to seconds within a scan. It is present both in awake humans as well as anesthetized macaque, although, as I said, there are recent studies that show that the level of variability or of volatility seems to go down with deeper levels of anesthesia. Whatever patterns you observe when you look at these short windows can be very different to what you see when you use the whole time series as a whole. And there's been some relationship to both these ideas like potentially getting drowsy or anatomical structural characteristics. And then there is this important concept of functional connectivity states, which is used in a lot of the literature that has happened after these initial studies. So let's keep looking a little more into studies that try to bring this link between the dynamics and potentially cognition. So one interesting study is this one by Tagliatucci, in which what they decide to do is, okay, well, one thing we could try to see is whether fluctuations in dynamic connectivity relate to sleep stages. You can just do it and guess whether you do it right or wrong, or you can go and try to compare it against the gold standard. The gold standard for scanning is usually having EEG recordings. So in this case, they did concurrent fMRI and EEG. They did the staging of the sleep stages based on the EEG. And with that, they train a classifier based on a subset of the fMRI data. And they look at how well that classifier works based only on the fMRI data. So the EEG data is just basically to provide labels for both training and for validation. But that doesn't go, there is no EEG information in the classifier. The classifier is supposed to perform its job just by looking at the different connectivity patterns across time. And I think they did on a window basis. Yes, windows of around 60 seconds to 4 minutes. And this is the results. Overall, they found an 80% accuracy. And here we have a couple of examples where you have the staging based on the EEG and the staying based on the fMRI. In this case, their tool of choice was a support vector matching. So again, it seems that, it contains information about fluctuations in sleep stages. We in our group decided to take a different approach. As I said, when you do these things in rest, it's really hard to know what to expect. So we decided to say, okay, let's impose some mental states by means of tasks, which is a really hard way to impose it. We're kind of setting up ourselves a little bit for success here, but it allows us to have a long scan in which we know what subjects are doing at specific blocks of time. So we decided to scan for around 25 minutes, and within those continuous 25 minutes, there was blocks of 3 minutes. For some of the blocks, the subjects were resting. For some of the blocks, the subjects were doing a two-back memory task in which they saw shapes, geometric shapes, and they had to press a button whenever the shape on the screen was the same as two-back. We had them doing a simple mathematical task where they have an operation on the top and two potential answers, and they had to choose the correct one, and a visual attention task in which they were looking at a video of a fish tank, and wherever there was a crosshair, they had to say whether it was on top of a specific type of fish or another. So here we have 3 very different cognitive tasks. For the 3 of them, we have behaviors, so we can know whether they were actually engaged on them or not. And we asked them to please, before entering the scanner, and they did one training run outside the scanner, to make sure they would come with a strategy of how they do the task and to keep it continuous during the scan, okay? So what we do then is we do something similar to what Elena Allen did. We define windows that we can play with many different windows duration, but windows that are definitely shorter than the duration of these 3 minutes. For each window, we compute the connectivity, of course, of this until after pre-processing of the data. And then basically for each window of time, we have 2 pieces of information. We know which task subjects were doing, but we also have these snapshots of connectivity. And the idea here was, okay, if these snapshots of connectivity contain information about what mental state, meaning which task the subjects were doing, we should be able to give these vectors to a clustering algorithm. And if I ask for 4 clusters, hopefully it will group them in ways that all the windows that belong to periods when the subjects were doing math go together, all the periods that belong to the windows when the subjects were doing memory go together, and so on. And again, the algorithm has 0 information about what tasks they were doing. It just takes these vectors of connectivity and it groups them. And it's me afterwards that compared to see whether that was the case or not. And basically what we saw is that it works quite well for most of the subjects, but we saw there were some subjects in which things work a little bit worse. I mean, the way to interpret this is just a way to look for level of agreement between a clustering made out of data and a clustering that you set up based on your expectation, accounting for potential agreement just by randomness. And basically anything in green looks that the clustering algorithm did very well, did a little bit worse, kind of bad, and definitely bad. Because we had behavioral measures for the subjects, we could see whether if this is really what we think, it should be that the guys who were really performing badly on the task, maybe they weren't even doing it or they didn't care that much, should be the guys that the algorithm classifies the worst. And that was the case. We had 3 guys that definitely did pretty bad in all measures, meaning percent correct responses, response times was the largest, and missing responses was the largest. And these guys were the guys for which the algorithm performed much worse. Here we see a significant correlation, but that goes away the moment you take out these 3 guys. So to which extent can these techniques give you very precise information about how well they're doing it? That is something we're currently investigating. Here, all we can say is that if you really weren't doing it, the algorithm seems to fail. And if you were doing it, the algorithm seems to work, which is good for what we were trying to test. Yeah, please, sorry. If you can take the mic that I've been told to please for the recording.
The question is, you showed that the classifier is able to detect the kind of tasks they're doing. Can you also use the same approach to identify subjects, or as opposed to looking to tasks?
So 2 things. The way this is done, it's unsupervised, so I cannot tell you which task they were doing. I can just tell you all these windows belong to the same sort of task. So that's the first thing. For me to tell you that now all these windows mean you were doing math, that would take an extra step that we didn't do. We could have tried to do a supervised classifier, some sort of support vector matching, and maybe we could have done that. So, but that was not what we did. Regarding the subjects, we didn't try it. I mean, there is studies by Emily Finn that shows that, but we haven't tried on this data. There is a study by Oliver sitting here that says that if you want to try, and this was all done within subject, if you want to go across subjects, for things to work, you have to account for subjects differences. That's one of the studies on the third slide for the special issue on neuroimage on dynamic connectivity. So you can find more about that there. Any more questions? Okay. So here is just another study. I think just two more, just to prove this point. Then I'll go into the bad ones too, so don't worry. I have both sides of the story. So this is a work by Thompson and colleagues back in 2013. And here what they decide to go with a very, very fine TR, just by basically acquiring one or two slices. I don't remember. And they look at whether the amount of anti-correlation between the default mode network and the task-positive network would allow you to predict how well subjects were going to perform in an upcoming trial. I don't remember. It's a psychomotor vigilance test. But there is a big range of studies that have looked at this. And the answer is yes. What they did is they divided their subjects in two groups. The guys that had really slow response times, I believe. Is that the slow? Yes. And the guys who had really fast response time. And they could find that there was a significant difference in the level of anti-correlation between these two networks. And there's been a lot of more work in this direction in recent years. And then once we say, okay, it seems here we have a new dimension in our data that potentially give us information about something that is related to how the brain is working, not just simply noise. So there's been some other groups that have decided to look whether here we can find also information about clinical questions. And in particular, this is a study in which they compare schizophrenics to normal controls. And what they found was that the amount of time, because that's one thing I've forgotten, once you have this framework of states, then there is a lot of things you can do. You can start talking about the amount of time subjects spend on a given state. You can start talking about trajectories that are common. Like, I don't know, in one group, it might be very common to see going from state 1 to 7 to 5, while on another group, that's not a common trajectory. So there are a lot of meta metrics that you can compute out of those frameworks. And what they saw in this case is that the amount of time that subjects spend on a given state, only for some of the states, you could actually find a difference between normals and between normals and schizophrenics, meaning that by opening this new dimension, there are things that you couldn't see should you be looking just as the stationary time series. This is work that requires much more confirmation, but this is another direction in which people is looking at that. But not everything is positive, and this is a study that I would recommend to all of you to read. It came out a few months ago. And basically what they claim here is that, hey, it's not just that. So a word of caution. So in their abstract, they very clearly say that by using simulations and different sets of fMRI data, they can confirm that imposed tasks can alter the correlation structure of ball activity. That should you be having your subjects doing different things, similar to what we did or similar to what Thompson did, things like that, you could expect that there will be dynamic changes in connectivity. But that they find that the observation of dynamic ball correlations during resting state are largely explained by sampling variability, and I will go into this in a second. But basically, we have our two ROIs, and we compute this sliding window of correlation. And we say, it changed. But that's our estimation. We don't really have for each of them a confidence interval. So we might be looking at something that is just noise. We need to do some statistical tests because otherwise we're just looking at an estimation. And it's important to add that statistical test. And should you not having, you might be seeing things that is just the result of sampling variability. And they claim that's one thing, head motion being the second one, and fluctuating sleep states being the third one. And for them, what they conclude is that aside from the preceding explanatory factors, a single correlation structure, meaning what you get out of the stationary analysis that we've been doing in the past, may adequately describe resting state as measured by fMRI. They do a lot of simulations. One that I find interesting to look at is this one, in which they have simulated data that is supposed to be statistically equal to real data, with the exception that they know is stationary from a statistical point of view, and real fMRI data. And when they go and they do these sort of k-means studies, they can find very similar states. So this is something to think about how much the methods by themselves may actually lead you to see things that you might overinterpret. That being said, I think one key thing to keep in mind that this is all about how you define dynamics, whether you're testing against some hypothesis. So I wouldn't take this as everything is wrong. I think it's important to read it. It's also important to keep in mind that they do this from a very specific description of what they mean by dynamics, which is a fourth order statistics called multivariate kurtosis. So again, a word of caution to think about what we're looking at and how we interpret it, but not definitely a way of saying that what we're looking at is all wrong, okay? So second set of conclusions before we go into the boring part of methods. Dynamic changes in connectivity and dose order scale seem to be good at reliably detecting different sleep stages, at discriminating between externally imposed mental states, and even at predicting task performance on the individual basis. There is a huge diversity on how everybody does those things. There are differences in acquisition, scan durations, TRs, window length, multiband versus not multiband, you name it. There are differences in preprocessing. There are differences. A lot of these methods require you to start by having a parcellation of the brain, because doing connectivity of every single voxel to every other voxel of the brain is very hard computationally. So as I will show you in a second, what ROIs you choose is going to potentially affect the results. What metrics you use to capture the dynamics, which methods. So it's really, really hard to actually do comparison across studies. And this is something that is a challenge for this field nowadays. That being said, some people are starting to explore the clinical applications. So you have both ends of the spectrum here. So any questions before I go into methods? Okay. So this is the paper I was mentioning. It's the latest review. I think it's a good start point. So there are two main lines of how to think about it. There is the line of a sliding window-based correlation analysis, with the idea being that this connectivity does fluctuate continuously across time somehow. And this is one way of capturing, perhaps not the best one, but it's a way of, that fits that whole idea that things are gonna evolve in a pseudo-smooth manner across time. And so basically, as we've done in the past, you have your time series, you create windows. For each of those windows, you compute a metric of connectivity. And so far, most of the studies, and I think almost everything I presented, that measure is Pearson correlation, but could be mutual information. Just pick your poison there. And with that, you end up with this matrix of connectivity or snapshots for the different windows. And then with them, you can either estimate functional connectivity states or something else. And then based on that, as I said, you can start computing those extra metrics like amount of dwell time for each state, trajectories, you name it. The other way to think about it is that actually what we see in connectivity is being mainly driven by a few instantaneous, but discrete events that happen and are very strong and happen only at very specific points. And actually those big guys, which big meaning above a threshold, and how you choose that threshold, it's a whole other wall, are the ones that drive everything else. And by focusing just on those guys, in a way that I will show you in a second, that's enough to analyze dynamics. Those are kind of the main two ways of thinking about this. Okay, so regarding a sliding window correlation, which as I say, is perhaps the most widely used, the idea being, you can do a stationary connectivity, but basically taking all your time series and the whole duration and computing a single matrix. Or you can do this kind of windows in which just for the first 60 seconds, you compute one. Then you move 2 seconds. So from second 2 to 62, you get another one, and so on until you get to the end. Many questions here. What window type to use? We can use rectangular windows, which would give the same weight to every single time point within that window. We can use tapered windows, so with a little bit less a little bit more protected against outliers at the beginning and at the end, and it might help. Or nobody tells that we cannot use a window that looks something like this, in the sense that to the more recent points to where I am now, I give it a lot of weight, and for things that come from the past, I give them less and less weight. And you can find studies with all of these options. What window length? Here is a trade-off, because if we use two short windows, and I will talk a little bit more in a second, we have very few samples, and therefore my estimates of correlation are going to be not very reliable. If I use very long windows, I'm maybe missing some really interesting phenomenon that happens at a shorter scale, and a few other things that I will tell you in a second. So that's not an easy choice. And what's the window step? We can, as I say, have a window of 60 seconds and then move it every TR. If my TR is 2 seconds, so every 2, or I can decide to have completely non-overlapping windows. If I have overlapping windows, then I have to be careful with whatever statistics I do later, because those are not independent windows. A window to the next, they're almost the same just by one data point, so things get complicated. So what are the pros of a sliding window analysis? It seems really easy to interpret, which seems being a big word there, okay? And it seems, or that we can say there's been studies that actually looks like it can capture phenomena that is potentially biologically and neuronally relevant. So those are good things. Now, it requires a series of a priori decisions, like the window length, the shape, blah, blah, blah. As I said, if you go too short, you might have unreliable estimates. If you go too long, you might impede observation, but the interpretation is a little bit more complex than it seems. And I'm going to go into this in a second. So this one, I'll just explain it in a second. So first of all, as I said, just because you see this variability doesn't mean that's there. You're sampling, so you have to make a statistical test. So one problem that this is a very good paper to read if you want to get a little bit into the the literature that tries to see some of the potential interpretational issues with a sliding window. They say that it's a problem to identify an observed value of a test statistic with its true underlying value. Just it's an estimate of that window, and it's an estimate of that window. We need to put confidence intervals, and we need to see that those confidence intervals actually are outside of what would be the stationary metric. So in a case like this, we could only claim that it is during this period and this period that actually you're away from that stationary value. Again, the idea, very, very simplified, but just having this idea that just because I see it moving, I have to do something else. And that something else means comparing against a null distribution. And that null distribution should be stationary, because what I'm trying to claim is the other idea, that it's non-stationary, but keep as much as possible all the other statistical characteristics of the data. And that's a little bit complicated. There are some methods and some ideas, and this is a good point to start. Another thing to be careful is that when I suddenly cut my data in chunks, I have to be careful about what are the frequencies of the signals. Because if I were to take windows that are too short to cover the whole fluctuation of if my signal contains fluctuation that are so slow that they don't fit completely into the window, that will lead me to having spurious fluctuations in my estimate of correlation. So here we have an example, very simple example of two sinusoids. They're off by a given offset, and that means that their connectivity is this one. It's not 1 because they're not sitting on top of each other, it's 0.6. But this is stationary thing. I mean, they're always as away as time progresses. What you will see here when I start playing the video, when the window length is too short to actually include a whole period of this, my estimate of the connectivity varies very, very much and a lot away from what is the true connectivity. And I might go and interpret that this is like there is a lot of dynamics, but that's not the case. And it's only when the window hits this point that we can see here in black that my estimate, I mean, it's still a little bit noisy, but really sits around the true estimate. So one important thing to do with my data to avoid this confound is to always make sure that prior to doing the sliding window analysis, I filter it, a high-pass filter, so everything that is too slow to not fit in the window gets removed. That will help with some of the confounds to a given extent. Here we have examples of how, using the same data, for 3 different window lengths. This one doesn't matter because this is the appropriate one, but here we have just keeping the same filter in all 3 cases, just reducing the window length and keeping the same filter, but reducing the window length, but also doing the appropriate filtering. We can see that by doing the appropriate filtering, we're dampening quite a bit this dynamic behavior, but we're dampening well. I mean, the idea is that all these things are not truly there. It's the result of this artifact, okay? Yeah.
You showed that figure before, that two perfect sinusoidal
Yeah.
signals. The correlation between them can vary from +1 to -1 depending on how lagged they are.
But from a statistic point of view, the two signals are, those two processes are stationary in their relationship because they have the same variance, they have the same mean, and they're just going together with the same dephasing.
Right, what I mean is that depending on how much they are out of phase, if you compute a whole static correlation, it's going to be different.
Yeah, of course. I mean, yes, that was my blue line, my green line.
And in some circumstance, I think if you have a half a pi difference, then that correlation would be set, but for the whole thing, 0. But if you do dynamic, then you see going from
Yeah,
minus
exactly.
one.
That's the.
Again, this is not explaining anything exciting. It's just
That's the point. That's exactly the point. To be careful, because you might see some dynamics that are not real. It's just a result of how you're doing things. Exactly. That's an artifact. Yes. Sorry if when I explain it, you may have thought I meant that that's interesting. No. This is a problem that we have to take care of. Yes. In fact, taking it into account in our own experience, for example, when we were doing this study, we can see that if we just apply the same filtering independently of the window for the shortest windows, which is where this would become more of a problem, the classification starts to go down. While by doing the appropriate filtering, we always keep it up here. So again, it's not gonna kill everything. I mean, they fall each other, but it's something to be careful because it's gonna introduce artifactual spurious fluctuations on the correlation traces. Yes, definitely. The other thing to think about is that the longer the window, what people say many times, well, the longer the window, the less the observed variability. And that's because that's how it should be. Because when you do a sliding window, it actually adds as a low-pass filter with a cutoff frequency of this on your observed connectivity. So what that means is that when you fix this, every fluctuation on the relationship between the sine series that is faster than this is going to be very much dampened. So when you're setting your window length, you're also kind of setting yourself to what's the maximum frequency of the dynamic phenomena that you can observe. That's why it's so important to pick the window appropriately. So here we have 2 time series that have been appropriately filtered. So that's why there is nothing below 1 divided by W. And what we can see here is that when we compute their connectivity in a very noisy estimate that is basically the pointwise multiplication, the fluctuation that we see in the connectivity, in that case, covers the whole range of frequencies. But when we do the sliding window correlation or the covariance, everything above here is being damped. And it's because this effect that actually the window does on our potential estimates of connectivity. So this is again a problem, something that we need to think about. So spurious fluctuations in dynamic connectivity can be limited by appropriate high-pass filtering. That was the first problem, but the remaining fluctuations will be low-pass filters. So not seeing something above that doesn't mean it wasn't there potentially. It just means you cannot see it. A word on parcellations. There's been several studies, and in general, when you're going to do this type of analysis, it seems to be a good idea to use a functionally based parcellation versus an anatomically based parcellation. That's just in case you're going to do these things. For more information, this is a good place to look. In our particular study, we decided to look at the spatial resolution of the parcellation. There are parcellations to spare, okay? I mean, every 2 weeks you get a new one. One we tend to use in our studies sometimes is the Craddock Atlas because it gives you contiguous ROIs and it gives you these ROIs at different levels of spatial detail. You can subdivide the brain in 100 ROIs, in 200, in 500. So it allows us to play with this. I'm not saying it's the best parcellation, but we've used it in several studies. And what we can see here is that if I use At too small parcellation, the classification works very badly, most likely because my ROIs are so big that I'm washing out effects of interest. I mean, I'm representing all this chunk of the brain by a single ROI. So anything that was important within there to many other within parts of the other ones is gone. And as I increase my number of ROIs, it seems to get better. Again, when you increase the number of ROIs, you're actually increasing the size of your feature space, and that can be a problem. In this case, we're using dimensionality reduction as an extra step, so that's why we might not be seeing that lower end of the curves at the other side. We also decided to look at the clustering algorithm, and we saw that k-means in our particular case performs better than hierarchical clustering. This is not just to tell you to use k-means. This is to tell you that there is a lot of methodological decisions that you take, and that they might influence how well that relationship between observed dynamics and actual effects of interest, meaning cognitive or clinical, you might see. And I don't have a pipeline of what's the right thing to do yet. But there are other methods. I mean, because the sliding window has so many issues, there's been other methods proposed. So I just want to walk fast through a few of them. One of them is this thing called dynamic conditional correlation. This is a model taken from the financial literature. It basically allows you to compute an instantaneous value of the connectivity. Think of it that way, an instantaneous R, but based on some big models on the background. Actually, it's quite computationally expensive. And in this original study, with Martin Lindquist proposed it as a potential use to study dynamic connectivity, they tested on different types of data. They tested it on data in which they imposed this slow fluctuation in connectivity, and then they went ahead and connected estimates of the actual connectivity using different methods. And in all cases, they saw that their method shows the least square errors different between the expected and the real one. So it seems to outperform the other ones. It's not like those ones will give you no information, okay? It's just that there are better ways to do it, assumingly. They also tried to test it in a case in which you have a more discrete event, because we may have both types of dynamics. And in this case, they also saw that their method performs a little bit better or equally to the other ones. Another way to think about this, and this is also coming from the same group, is, okay, instead of... and there's been other cases in which what they do, they start with very short windows, and they let the window increase, and they test for stationarity using a statistical test. And basically say, okay, is my window still stationary? Yes. I make it longer. Yes, yes. When it's not, that's my window for this point. And then they move to the next. And you can end up with different windows of different length. Another way to think about it is this other technique in which they try to detect what they call temporal time points. The idea being you have a pattern of connectivity, and that's going to be constant for a while until there is this point in which the algorithm detects there's been a change in the pattern of connectivity. Then you have a new pattern that will stay for a while, and so on. And the algorithm is able to detect those change points and the associated configurations of connectivity in between. In this particular case, in this paper, you can find a lot of simulations, and then they went ahead and tested in this data set, which is an on-off design in which they have subjects. They tell them basically, look, you're going to go into the scanner, and there is a high likelihood that when you come out, you're going to have to give a speech in front of a panel of people. So for the first 2 minutes, they don't tell them any topic, nothing, they're just resting. At minute 2, they tell them, here is the topic. Start thinking about what you're going to say about this. And then around 2-3 minutes later, they tell them, well, you've been lucky, you're one of those who's not going to give the speech. So the idea being that you have 3 different levels of anxiety. And they applied their method to the data, and they found that in many cases, the first change point seems to agree with when we go from period 1 to period 2. But you can see here that this one is much more variable. Now, is that a problem with the method, or is that a problem that not everybody stops being anxious at the same time? It's hard to tell. Some limitations of this method is that because it's so computationally expensive, I think they say that anything with more than 100 ROI is hard to work with it. And it's not a method that, okay, we're not setting the window length, but you have to set a lot of other parameters. So, nothing comes for free. Now, let me just talk for the last 5 minutes on the other type of methods, the one that assume that there is a few discrete events that is what matter, because those are the ones that tell you everything about the dynamics. So in a sliding window, we assume that the spontaneous brain activity is characterized by slow, continuous evolving dynamics. Here, all the information is condensed in a few events. So what we can do, what was proposed, you pick your voxel in the posterior cingulate cortex or a seat. You have their time series. Sorry, you set a threshold, and then you go and look to the volume, just that volume of your data after preprocessing at these time points that are above that threshold. They all look like they will have red on the PCC, more or less, because they all have been selected because they have a high value of intensity in the time series in the PCC. And they all look kind of similar, but very different. I mean, partly it's a single time point, there is a lot of noise. So what they did is like they decided to average all of them. And the first thing they saw is that when you average all of them, you get a pattern of connectivity that actually is activity, because this is just average. But this pattern of activity looks a lot like the patterns of connectivity that you get when you look at the whole time series. But because all these guys are not the same, what they decided to do is we can take all these guys, the guys that are above the threshold, and apply some sort of clustering to come with these configurations that seems to appear related to this set. And this is what they call CAPs, co-activation patterns. So basically, they decide on a number of clusters, and then for each of them, they see when they happen. They don't need to be contiguous. So here is the red guys, you have them for a while here, and then a little bit later. And each of them, you can get a representative. And once they do this, they can go ahead and take any of the networks that you see on a stationary matter and subdivide it into these caps and see how much each of these caps contributes, how much each of these caps is actually equal to that overall, and what fraction of time each of them is present. What to do with this? I don't know. There are papers out there that are trying to look at that.
How does it relate to event-related, and when you do a task with this method? Because it seems that the events will influence the connectivity.
Well, it can be applied to that, but the idea being also that during rest there are these big events. And are those big events that maybe it's because you're doing some mental tasks, you're just it's not being imposed on you externally. Now, because you don't know where they're coming from. And there are some other methods like paradigm-free mapping and total activation that actually will ensure that those things actually follow an hemodynamic response. So you might know they're truly hemodynamic. Those are truly hemodynamic discrete events. But how do you interpret them? I mean, the claim here is that those explain the dynamic behavior, and that just by looking at those, we can understand the whole dynamic phenomena. But how to make more of them, I mean, maybe there are some studies, I just don't know.
When you impose the task, do these events correspond?
Of course. Yeah, if you have an event related, you would see them like this, yes. Yes, definitely.
So that method would be just for pure resting state, not resting state.
Yeah, you can do it for resting state. The other thing you can do, and this is something I'm working with some collaborators, is not with this, but when you look at the convolution methods, for example, some people is trying to, when you do resting state on people with epilepsy, and you don't know whether they're having an interictal event or not. The convolution methods may allow you to go and find those events having no a priori information about when the task, meaning the interictal event, happened. Or they may allow you, even though you're doing a task, when you do a task, you go with your paradigm about the explicit moments when the task was happening. But there are a lot of other things happening, so it allows you to do that deeper exploration, but it becomes harder to interpret. And then just for your joy, here are a lot of other methods, mostly taken from the pretty review. There are time frequency analysis, which open a whole other dimension into your data. There are methods based on multiplication of temporal derivatives, hidden Markov models, dynamic graph analysis, you name it. It's exploding field with a lot of options, and it's hard to choose. That's all, unfortunately, what I can tell. But just some general conclusions. As I say, it seems there's this rich dynamic behavior that captures our attention, and that's why we are all spending so much time in trying to see what it means and what's the best way of getting a handle into it. There are these short-term patterns that seems to be reoccurring in time, and that to a given extent, some of them we can relate them to tasks. Evidence suggests, yes, what I just said, sorry. And some people is exploring, as I said, this as a potential biomarker for clinical conditions. Now, we need to better understand which methods actually capture these biologically or neurally relevant functional connectivity dynamics best. A lot of those methods is going to give you a picture of dynamics, and that's not enough to claim that what you're seeing is neuronally or biologically relevant. So that's important. That's also not telling you it's wrong. It means you need to think about it more. It is unclear to what extent dynamic functional connectivity was conceptualized as this multi-stable, slow changing thing, or just these discrete events. So this is something still open, or maybe both views are meaningful. Just some of them is better to study A, and some of it is better to study B. And then the study of dynamics opened this question about how this concept of network, the one we're used to see, is really something real or something elusive, knowing that they completely break down and fall apart for long periods of time. And with that, thank you. And if you have any questions, please go ahead. Oliver?
Yeah, I have a question. I know you do like physiological noise regression all the time, but have you looked at before and after the physiological noise removal, what dynamics will change?
No, I haven't. So the question is about whether how does physiological noise may affect the dynamics that you see. So I tend to do it as much as I can. I may miss sometimes. But physiological noise, overall, though some people may claim that, of course, when you breathe, so there is some neuronal activation associated. But overall, you can think of it as a confound. So I don't see why you wouldn't want to remove it on the first place and look at what happens if you don't. I think it's a good practice to remove it. There are many different ways. There are ways in which you can remove it when you have these external traces of cardiac and respiratory functions, and there are ways in which you can remove it even if you don't have them. So I would advise to do it all the time for this.
Sorry, just for the monkey study that you mentioned earlier, that they put monkeys into anesthesia, and do you think that could be attributed to when you put the monkeys into anesthesia, then the physiological noise, like the fluctuation, will be less, then lead to a decrease of the function dynamic?
I wouldn't attribute it to that, but I mean, it's something that could be checked. Definitely when they're in anesthesia, they're usually on a respiration machine, so it's a very steady, at least respiration, and it's easy to remove it, the artifact of that. I don't remember what they did in this particular study regarding that. Thank you.